Hi, my name is Karen and I'm a sophomore at the Marine Academy of Technology and Environmental Science. This is my study on the effects of ultraviolet radiation and temperature on biodegradation of polyethylene terephthalate using Bacillus subtilis. The objective of this study was to explore the effects of ultraviolet radiation and temperature on polyethylene terephthalate degradation while maintaining the health of a pure strain of Bacillus subtilis. It was hypothesized that using UV radiation and Bacillus subtilis biodegradation at a temperature of 50 degrees Celsius, PET will exhibit significant loss of mass while the bacteria remains viable. $7.8 billion tons of plastic are produced globally. Polyethylene terephthalate is the most widely used because it is durable and lightweight. It is commonly used in water bottles and food packaging. Production has increased during the COVID-19 pandemic for personal protection equipment such as face masks. Unfortunately, PET can take up to 1,000 years to fully decompose and is accumulating in ecosystems and harming wildlife. Shown here is the structure of PET monomer, where N is the number of repetitions. It is a stiff molecule and lacks segmental mobility and can withstand a wide range of temperatures. One solution is microbial bioremediation, which is the use of microbes to decontaminate ecosystems by breaking down pollutants. Using biotic components in situ avoids the need for harsh chemicals and costly pollutant extractions. It can be used to degrade plastic at the microscopic scale, which is especially important when dealing with microplastics. One example of a microbe is Bacillus subtilis, also known as hay bacillus. It is commonly found in soil, vegetation, the human gut microbiome, and aquatic environments. It can naturally biodegrade PET, using it as its main energy source and decomposing its molecules completely. Using naturally occurring strains of bacteria opposed to genetically modified organisms is important to be able to track the metabolic pathways of the PET. Bacillus subtilis can withstand ultraviolet radiation and high temperatures. The three types of UV radiation are UVA, UVB, and UVC. UVC has the shortest wavelength and can break down the hydrogen and carbon bonds in the PET molecules. In this study, PET biodegradation by Bacillus subtilis was measured under varying temperatures and exposure to UVC radiation. PET mass was analyzed to measure biodegradation. Turbidity and bacterial area coverage were used to measure bacterial viability. Many current studies on PET biodegradation focus on gene editing and enzymatic processes. However, this study attempts to explore factors affecting degradation efficiency while maintaining the health of a naturally occurring bacterial strain. In this study, the sample size was 60, which was split into 6 groups of 10. Group letters denote exposure to UV radiation, while group numbers denote incubation temperature. First, I reactivated the bacterial cultures, plated them on agar, and took pictures of the plates. Next, I inoculated the samples in Luria broth and vials and recorded 3 turbidity measurements for each sample. Simultaneously to the bacteria preparation, I pre-treated the PET strips with UV exposure and recorded the mass before and after. Then I put 1 PET strip in each sample vial for biodegradation and incubated the groups at the specified temperature. After incubation, I extracted, sterilized, and masked the PET strips. Next, I measured the turbidity of each sample. Then, I plated 3 random samples from each group on agar and took pictures before and after incubation. Mass of PET strips was used to measure degradation. A t-test was used to compare mass before and after UV exposure. There was a 0.3% decrease. However, no significance was established between mean PET mass before and after UV exposure. A t-test was conducted to compare before and after turbidity before and after mass measurements for each group. Group 1 exhibited no significance between mean PET mass before and after incubation, while groups 2 and 3 did. Percent change in PET mass was calculated for each sample. No significance was calculated between mean percent change at each incubation temperature. Turbidity was used to measure bacterial viability in liquid suspension. Significance was established between mean turbidity before and after incubation. The difference in turbidity before and after incubation was calculated. Group 1 indicated a significantly larger difference in turbidity than groups 2 and 3. Bacterial coverage was calculated using ImageJ software and was used as a parameter to measure bacterial viability. No significance was established between mean percent change for each group, before and after UV exposure. Overall, UV radiation and temperature did not affect PET degradation during this experiment. UV radiation did not cause a significant change in mean PET mass. The increase in mass after incubation was likely caused by the formation of a biofilm, which was not removed during the sterilization process. Scanning electron microscopy could be used to analyze surface area changes such as biofilm formation and degradation. All incubation temperatures showed significant differences before and after incubation, indicating that the bacteria remained viable. Groups 2 and 3 displayed significantly less bacterial growth in liquid suspension because of the increased temperature. However, the bacteria remained viable because no threshold was reached in which the bacteria could not survive. Bacterial coverage did not significantly change during UVC exposure, indicating that Bacillus subtilis remained viable. Shown here is an example of coverage before and after UV exposure, indicating that Bacillus subtilis remained viable. Health of Bacillus subtilis must be maintained during UVC exposure to continue the biodegradation process. Further studies on PET biodegradation would be necessary to explore other environmental factors affecting the degradation process. To conclude, UV radiation and temperature did not significantly affect biodegradation of PET. Underlying factors that could have affected the results of this study include molecular changes of the PET monomer and the formation of a biofilm on the surface of the PET. This could be analyzed further using scanning electron microscopy. Bacillus subtilis remained viable after the biodegradation stage at all temperatures and exposure to UV radiation. It is important to note that the controlled environment in which this study was conducted does not accurately reflect biodegradation conditions in the natural environment. Thank you for listening to this presentation. If you have any questions, please feel free to contact me. Have a great day. Thank you for your time.